Introduction to genetics. Okay, genetics is a branch of science which deals with heredity and variation. Heredity and variation. So genetics is a branch of science which deals with heredity and variation. Now, what is heredity? Heredity is passing of traits from parents to offsprings. As a result, human beings give birth to human. A mango plant give birth to mango plant. So that passing of traits from parent to offsprings, and in which offsprings are very similar to that of the parents, that is heredity. Next comes variation. Variation is the differences among the offsprings. Two human baby, they will not be same even though they are of the same parents. So two brothers, they would be dissimilar, say dissimilar in height or their what you call voice, eye color, skin color. So they will be dissimilar. So a lot of dissimilarities would be there. So that is variation. The study of this heredity and variation is what we call genetics. When we study genetics, basically we can divide it into pre-Mendelian, Mendelian, then Mendelian genetics, Mendelian, and then post-Mendelian, post-Mendelian genetics. Okay. Now, pre-Mendelian genetics is a very old concept. Say the first half of human understanding of say living organisms giving birth to living organisms. So in that, the most dominant was that there was spontaneous generation. Okay, spontaneous generation. This is the pre-Mendelian concept. First, this was the belief that organisms arise spontaneously from non-living matter. So from non-living matter, this is also called as what you call abiogenesis. Abiogenesis, and these were the beliefs of all the great thinkers like Aristotle, Plato. They all thought that living organisms arise spontaneously from non-living matter. Example they gave to suppose the maggots, say the maggots or the worms, no, which will appear in say decaying meat. They will appear in the decaying meat, or there would be growth of microorganisms or say algae in stagnant water. So there they believed that non-living matter spontaneously gave birth to living organisms. Then this was first challenged by this person called uh, Reddy in 1668. Okay, Reddy. His view was that for life to come, say some organisms would be required. That means he said that spontaneously life cannot arise. There is no spontaneous origin. So for him, what he said that some amount of pre-existing life is required for the organisms to survive, or organisms to come into life. Pre-existing life is required for new organisms. Okay, so this is Reddy's view, and he what he did, uh, he provided the evidence of some maggots appearing in meat, but when that meat was covered, uh, then flies would not be able to enter, and if flies would not be able to enter, there will be no maggots arising on the meat. So this is what is said, and this was further uh, proved or further elaborated by Spallanzani. Spallanzani, what he did, he took a meat broth. He took meat broth, he heated it, and after that he sealed the glass bottle. Once that was sealed, then what will happen? So then no flies can enter. So no flies can enter means there were no maggots in the meat broth. So he said that for life to appear, pre-existing life is required. Okay. But then what the original thinkers or their followers of original thinkers said, that since you have sealed it, you have heated it, so basically you have killed the vital force. Okay, you have killed the vital force, and hence life is not there. So ultimately from here again it returned back to spontaneous generation. But then this great person, we can remember Pasteur, he did an experiment, or he devised a structure of this nature, in which what he did was he took a similar structure like this. Okay, but then he made a tube like that, no, so something like this. So this allowed air to enter. This was the meat broth which it was heated. This was the culture, and structure of this nature was there. When the design was made like this, so what happened, any dust or any flies, they could not enter over here. So whatever things entered would simply by gravity will fall over here. This place will be neat and clean, and here hence maggots would not be seen. So then he allowed, or he showed, that air is also entering. Okay, and he is not killing the vital force. So but still we are not finding the flies. So this Pasteur, for the first time, without any what do you call without any doubt, he proved this theory of biogenesis. Biogenesis, that is, for life to appear, pre-existing life is required. So this is what we also call as omnivivum ex vivo. All life comes from pre-existing life. So this was the pre-Mendelian concept. So at least in the pre-Mendelian concept, we have understood that for life to appear, pre-existing life is required. Then comes this Mendelian concept, in which he talks about what? He talks about your uh, heredity, and he talks also about why the variation could be there. So here he is laying the foundation of genetics. And then whatever Mendel said, all these things are not true, as we will see, and those which are exceptions to Mendel, which afterwards, after study, lot of changes has been brought, those will come under post Mendelian concept. So this is the whole genetics which we will be talking about. Now let's move to the Mendelian genetics. So for understanding Mendelian genetics, you have to understand certain terms. Now first term is alleles. Okay, alleles basically are the different form of a gene that determine alternate traits of a characteristics. So we already know, suppose say an allele always occurs in pairs. So this is, we know, suppose this character is tall. Now here we can say that one gene has come from the father, one gene has come from the father, say one has come from the mother. So now each one is an allele. So each one we can call it as an allele. Okay. So always in a character we will find that there are two alleles. Now genotype. What is the genotype? Genotype is the allelic construction for a particular trait or character is called as a genotype. Example for tall, say tall could be capital T could be tall. So this is the genotype. Or capital T small t also could be tall. So this is another genotype. So genotype is the genetic composition. So genetic composition, but here both the genetic composition is showing the character tall. Phenotype. Phenotype is what is the expressed character or outward physical character. So in genotype or genetic composition we cannot see it, but phenotype is what we will observe. What we will observe is the phenotype. So whatever calculations, whatever structures of the genes, alleles we make, these are all based on the phenotypic observations, or the observations which we can see. Then another two terms we will find: dominant and recessive. Dominant, we all know, suppose say here two alleles are present. So here small t one allele, capital T one allele, but still we are finding that the character expressed which we can see is tall. So then we can say that capital T is a dominant allele. This is dominant. And small t, which is not able to express itself in presence of dominant, so this will be recessive allele. So alleles would be dominant or recessive, and afterwards we will see that whatever characters which are expressed in the F1 generation, they are, this is the dominant character. So character expressed in the F1 is your dominant character. Some other terms, homozygous and heterozygous. Homozygous means when alleles are same, both are same. So capital T or small t, this is homozygous. Heterozygous would be when the allele pairs are different. So here suppose capital T and small t, this is heterozygous. So two alleles are different is heterozygous. Next two terms we will see, pure and hybrid. Pure means which on selfing, basically we can understand that we can also say that the homozygous, this homozygous, they are pure. Okay, means that means on selfing means if you self these two, what is the type you will get? This from here again you will get the same character. This if you cross, what you will get? Small t, this is small t, and then this small t, small t. But you consider a case of this nature, there you will find that there could be combinations of capital T and capital T small t or small t small t. So you are not finding the parental character alone, you are finding some other characters also coming out. So then this will be your hybrid, this will be your hybrid. So that means on selfing, if it is showing more than its character, more than the character which it has, it is showing some other characters, then that is hybrid. And on selfing, if it is showing the same character as that of the parent, then that is pure. Next is monohybrid cross and dihybrid cross. Monohybrid means when you consider only one character for a cross, one character. So suppose I consider the character height. So height, if I consider which is could be capital T or small t, something like that. But the character considered is height. So one character. So then that will be your monohybrid cross. Monohybrid. But a cross in which we are considering two characters. Say suppose here common one was say seed color and say seed shape. Seed shape. So two characters we have considered, seed color and seed shape. So suppose seed color is seed shape is suppose round and seed color is yellow. So you are considering two characters. So then this is dihybrid cross. Now when we are talking about Mendelian genetics, we have to understand first about Gregor Johann Mendel, isn't it? Now, Gregor Johann Mendel, he was an Austrian monk born in Czech Republic in 1822. He came from a very humble background. As we can see, he's a peasant farmer, son of peasant farmer, and he studied theology and was ordained as a priest of Order Saint Augustine. Went to the University of Vienna, where he studied botany and learned the scientific methods. Worked with pure lines of peace for 8 years. And prior to Mendel, the heredity was regarded as a blending process. Okay, blending process means uh, whatever will be the characters of the parents. So suppose this is a father's character and there will be mother's character, isn't it? So the child will have mixing of these characters of fathers and mothers character will mix and it will be shown on the child. So this was the earlier concept. This is what we call as blending inheritance. That means both will mix and then that will be shown in the child. So some characters of father, some will be mother, that will be shown. But Mendel showed that it is not blending. So what he showed, we will see that. But here one very interesting thing I want to say you, that what do you think, was Mendel successful in his experiments? Because before Mendel also similar experiments were done by many other scientists, but they were not successful. But Mendel was successful, and the reason which we can ascertain to Mendel why he was successful, because Mendel chose pea plant. Now, why choosing pea plant made him successful? So pea plants are all homozygous. Very important, we have to understand. Pea plants are all homozygous in nature. Now you might ask, why pea plants are homozygous in nature? So here you have to understand that pea plants were inbreeding in nature. Pea plants were inbreeding. Okay, inbreeding in nature. Inbreeding. So what happens? Inbreeding means, that means if this is the flower, suppose this is anther, this is stigma, so they will do the breeding of, means breeding within themselves. That is inbreeding. Now, inbreeding leads to homozygosity. Okay, inbreeding leads to homozygosity. How? Just very quickly, if I want to show you, suppose if I take something like this, a cross like this. Okay, so what you get, say capital T, 2 capital T, small t, and small t, small t. Okay, if I break it into percentage, I can say that this is 25%, this is 50%, and this is again 25%. Now, since they are inbreeding in nature, so capital T, if they inbreed, again we will get all capital T. If this inbreed, what we will get again? We will get three things, isn't it? Capital T, then 2 capital T, small t, and then small t, small t. And here no problem, it would be again small t, small t. Now see, this 25% means all 25% will be same. It will come as it is, because inbreeding between them. Here this 50% got divided, isn't it? How it got divided? Say this could be 12.5. This will be 12.5, and this will be 25%. Yes. So now see, only first generation, if I do 25 plus 12.5, how much it will be? 20, 30, 37.5. So 37.5 homozygosity has occurred. 37.5. And if this, you can understand, if this continues, this I have just shown you for say the second generation, and if this continues for thousands of years, because pea plants were born before Mendel, lot before, so it went, this process went for thousands of years, isn't it? Thousands of years. As a result, whatever characters Mendel got, they were all of this nature. All were homozygous in nature. They were all homozygous in nature, isn't it? So that means the round which they got, it would be all of this nature. Wrinkled will be homozygous. Yellow, homozygous. Green, homozygous. Yes. Purple color, homozygous. White, homozygous. So the parental characters were, this is very important, the parental characters which Mendel considered were all homozygous. They were all homozygous. That is why he could come to a conclusion. So that is the main thing which we can understand. He was lucky. He was living in a holy place, the church, and might be the God was helping him select that plant whose all parental characters were homozygous. So when he studied it, he studied 7 traits. So these are the traits which we can say. So this is, say, number 1, say seed shape, and this is seed color, then this is the flower color, purple and white. This is your pod color, okay. Then this is your pod shape means either it could be inflated or it could be constricted. That means you can understand that seeds are present. No, how many seeds are present? Constricted with the seeds. Then your what do you call? This is flower position. Flower position is one is axial. Axial means it is coming to the axis. So axial, and the other one is terminal, at the tip. At the tip it is coming. So terminal, axial and terminal. And this is, say, 6, and then this, though we commonly know, this is dwarf variety, and that is the tall variety. So 7. These are the 7 characters which Mendel studied. Next, let us move to the principles of inheritance. So the video would be very long. So next video I will be starting from principles of inheritance. Thank you.